Well, thank you everyone for inviting me to present today. So there is something that's not showing up. It should be showing. Oh, there. Okay, so there's a little presentation there. So my name is Guillaume Butler-Laporte from the Division of Infectious Disease at McGill University. Thank you for inviting me. I still have fond memories of my time, a few occasions that I went to Bristol to actually present at some point at your university 2-3 years ago on another topic. But yeah, so this is the very typical Bristol view from a tourist perspective at the very least. But so yeah, very happy to present today. You've noticed that from the Division of Infectious Disease, so a bit off topic clinically from what you guys do, but I was asked to present basically our recent research on how we used basically genetic epidemiology based on biobank scale genomics to try to treat a patient of mine that I encountered very early on in my practice as an infectious disease physician with a monogenic disorder of immunity, and for which we had essentially gone through all the different lines of treatment for this. And so I'm going to present you essentially chronologically what we did as we first encountered the patient and then brought this to the lab, and then back to him with what we thought was going to be a therapy that could maybe work, and then we'll see if that worked, and then we'll try to speak about the greater question about this and how this can be maybe useful for you guys in oncology. So this was the patient. Basically, also, I'm a genetic epidemiologist. I know I'm probably speaking to a broad audience, so I make no real assumptions about your knowledge of genetics. If you have more either clinical or technical question about genomics or genetics, please feel free to email me. I'll be really happy to discuss this with you, or ask a question, of course. So the patient I encountered about 6 months into my clinical practice, inheriting this from a more senior physician who thought, hey, that's going to be a challenging case for you. Was a 39-year-old man, European ancestry, carried 2 rare pathogenic mutations of the CARD9 gene. CARD9, I'm told, is actually perhaps important in oncology and cell proliferation, but in infectious disease, it's mostly a gene that if you're deficient of it, you are inherently susceptible to all kinds of fungal infection, mainly candida, so yeast infections, but also, as what's the case of this patient, dermatophytosis, so dermatophyte infection, which I'll explain a bit what it is later on. But basically, he had chronic skin infection for the last 20 years to different fungi of the species Trichophyton. Initially, that was successfully treated with months-long course of itraconazole way before I had encountered the patient. He was then put on chronic suppressive treatment because there's not much more to do for these patients, unfortunately. But as is often the case with chronic suppression of infectious disease, antimicrobial resistance starts to appear and efficacy started to wane for him, unfortunately. So just a little reminder, I won't dwell on this too much, but dermatophytes, they're basically a class of infection, of fungal infection, and what characterizes them is that they thrive on keratin, which is a protein found in skin, hair, and nails. And you probably have heard about it more through the expression athlete's foot, ringworm, and jock itch. Those are dermatophytes. It's a very broad class of fungi, but they all share this characteristic that they thrive on keratin. So this was actually the patient. Of course, all the pictures I present today was with the patient's consent. Basically, he had these plaques all over the body. I'm only showing the shoulders because those are the, oh, someone talking, sorry. No. I'm only showing the shoulders because those were the biggest plaques. Those were the more symptomatic, but he had it across the body and other places in the back, in the genital area, and the legs, and unfortunately also on the toenails. What you can't really, I mean, you can definitely appreciate that there's a difference in color, but what we can't really appreciate too well is that it's very scaly. That's going to be important when we're going to assess the response to treatment. And also, you can't appreciate like the itchiness, the pruritus, how like the discomfort associated with this type of plaque that he's had to live through for a very long time. And to be clear, like a plaque of dermatophyte, which was proving on pathology and on culture and microbiology, that is this big is just, I mean, you need basically some sort of inborn error of immunity to have this because you never see this in a regular patient. And so, yeah, we had essentially been at the end of the line in terms of usual therapeutic approach, which was basically antimicrobial therapy. If you know a bit about card9 deficiency, you of course know that there are other approaches, like including interferon and other therapies, but they have a lot of side effects. Interferon therapy can be very nasty over the long term, so we tried to avoid those. And so basically asked myself, well, I'm a genetic epidemiologist. Maybe there's something I can do in terms of bioinformatics or in our biobanks and our data set that we can maybe bring back to this patient. And so this got me to think about this fairly famous by now in genetics kind of spectrum of diseases that we study in genetics. This patient clearly, this is a famous picture that's been cited and used and reused in many presentations and papers around the world. But this is basically the spectrum of disease that we study in genetics. This patient definitely had rare alleles. To give you an idea, the frequency of the pathogenic variants he was carrying at card9 were with the order of less than frequency of less than 0.000001% in European populations, and so extremely rare. It was definitely causing a Mendelian disease, so a card9 deficiency. Admittedly, he had a milder phenotype than others, but it was still a Mendelian monogenic disease. Whereas in most of genetic epidemiology and in biobank scale genetic epidemiology that I use, we're looking mostly at the bottom right there, in case you can't see my mouse, the common variants implicated in common disease. Dermatophytosis is a very common disease. About 20% of the world population has it at any given time. Or the lower frequency or intermediate frequency variants with intermediate effects. So this is kind of where this diagonal there is what we study in human genetics, but really it's all part of one spectrum. And if you really think about it, you start to think, we're all on that spectrum genetically, and the therapeutic targets that we can find on one end, there's no reason that we can't try to apply them on the other end. And so that's what we set out to do. So basically, the big problem, of course, with genetic epidemiology of common disease with common variant is that the effect sizes are very small. And so as you can imagine, if you're going to study them and find anything meaningful that you can translate into a therapeutic target, you're going to need gigantic sample sizes. And this is the major breakthrough in human genetics, I think. It's not really scientific, it's almost more logistic and engineering, but this is still the major breakthrough in the last 10 years. It's just the sheer amount of data that's now available to researchers. And so we managed to basically from different sources, either to which we had primary access or that some of the summary statistics were being shared openly so we could use them and meta-analyze them into a larger analysis. But basically we managed to tally up a total of about 240,000 cases of dermatophyte. This is both like all-encompassing, so hair, nail, skin, and close to 2 million controls. And to give you an idea, so fairly admixed sample, like we got all of the major biobanks. So in the UK, you're probably familiar with the UK Biobank, which is 95% European, but 500,000 participants. We also had access now to all of us in the United States with about 400,000 participants, more even split Black Americans and Europeans. We also used the Million Veteran Program, which is about 600,000 participants, also a bit more mixed, but there's also Taiwan Biobank, FinGen in Finland. So we really dug up and spoke to researchers everywhere to try to dig up data from all available ancestry to really pump up those numbers, to be able to kind of to really get strong, robust signal, even for genetic loci where the effect size might not be that big. And so this is what we ended up doing. And this is the famous plot you see in those genome-wide association studies, which I'm going to explain because it, I think, very neatly summarizes what our findings were. Basically, this is called a Manhattan plot just because it resembles a city skyline, nothing special there. But the idea is that each little point is a different common variant, so an allele frequency of at least 0.5%. So nothing really rare, something that you could find fairly easily in like the UK Biobank, for example. And so each point is a different a genetic variant, most of the time a SNP, a single nucleotide polymorphism. And they're spread out across the chromosomes, of course, 1 to 23. And then the idea here is that the y-axis represents the p-value. So the higher the point, the smaller the p-value. So to give you an idea, the line was written 60 there. This variant would have a p-value of its association with the trait of 10 to the minus 60. So basically, those are all p-values of the logistic regressions between the different SNPs, the different variants, which are represented by the dots, and dermatophytosis through the case control association that I presented above. So this is basically just logistic regression throughout the genome with the p-values. You'll see here, faintly maybe, but a dashed horizontal line, which for various historical purposes, we now refer to commonly as a genome-wide significant threshold, which is basically in GWAS and genetic epi, our line for Bonferroni correction. It's a bit more complicated than that, but basically it's our association with multiple testing for p-values. And so what we'd like to see is these little columns of variants there. So a big one there in chromosome 16, another big one in chromosome 8, another big one in chromosome 6, which is DHLA most likely, in case you're wondering. And so every little column there that crosses that line, we consider this to be a likely region where there is genetic association with the trait. Now, this is all about p-values. Doesn't tell you anything about the betas. And so if you're wondering, I'm going to show them a bit quickly here. I don't want to dwell on it too much. The point is basically that the genetic effects on the betas are roughly the same across the population, and that as expected, hopefully you can see this, as expected, the sample sizes are not very big. So those are the betas on the logistic scale. So if you want to get the odds ratios, you need to exponentiate this. And basically we rank that from the top left to the bottom right into like the most significant in terms of p-value to the least significant. And so as you can see, like we have massive sample size, and so we can pick up with a p-value of 10 to the minus 60, 10 to the minus 70, an effect size of 0.05 of beta, which is about 1.06 of odds ratios. But this effect size is very homogeneous across the different ancestries and the different cohorts. And so this is very robust. This is very reassuring. And you can see this throughout the entire loci, the entire GWAS. So this is very reassuring, but yeah, smaller sample sizes, but very robust, right? We have very homogeneous signal throughout the different cohort. And so in the last 10 years, from this, there's been a lot of groups in different diseases that have been able to use this very robust data to translate this into treatment targets. And this is what we will hopefully do in the rest of the presentation. So now that I've showed you that the p-value is very small and that the betas were most likely robust and reproducible, even though they were not necessarily that big compared to like rare variants underlying monogenic traits, the big question is still, anything interesting that we found at these loci, right? Because if we can't find anything that's clinically actionable, well, I mean, it might be scientifically fun, but not much I can bring back to the patient. And indeed, I will recognize this because this is a common criticism of genome-wide association studies. Sometimes results can be a bit perplexing and can be difficult to actually decorticate. And I'll give you an example here, actually, at the most significant locus in terms of p-value, at least, that we found. So I highlight here, surrounded by this green box. And just imagine, I'm going to now switch it to a locus, what we call a locus zoom, which is essentially the same y-axis, but the x-axis is going to be stretched out only to show this little region there. And it's going to look something like this. Okay, so the y-axis hasn't changed. I'm sorry if it's a bit blurry. Y-axis hasn't changed, but you can see that we zoomed in on it so that it doesn't look like a straight column there. It's actually stretched out a bit, and you can actually appreciate the different variants that make up this large column. And so, as I said, the odds ratio quite small, 1.06, but the confidence interval is basically 1.06. And this is often what we get in genome-wide association study. We get basically a strong signal that's reproducible, and then you look down at the protein coding gene in that vicinity, and you're like, oh boy, like 20 of them. Which one is actually causal? Which one actually underlies that disease? Which one is actually meaningfully actionable? Not that easy. Now, in this case, I must say after 6 months, I'm fairly convinced that it's PRSS 53. We looked into all of those. We did a lot of dry lab analysis about this. I'm fairly confident it's PRSS 53, which is a gene which is responsible for curly hair, which is so it acts on the hair, so that makes sense in terms of the metaphytes. And as you'll see later, we're going to talk about retinoic acid. Retinoic acid actually it acts on PRSS 53 and can actually change hair consistency. But this is not something that after I was looking at the GWAS result that I could know right off the bat. Like there's definitely a lot more work that needs to be done in this to translate this into something that's actionable. Now, thankfully, sometimes things are a lot easier. So sometimes, as you'll see a bit later, there are situations where there's one peak there on locus zoom, and at the bottom is only one gene. In others, there's a ton of gene, but and so you panic a bit the first time you see this, but then you realize that all these genes starts with the prefix KRT, 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 KRT everywhere. And so these, in this specific situation, chromosome 12, these are all keratin protein coding genes. So they all encode from some form of keratin, which is nice and actually a very good positive control that we found that different variation of the keratin protein may lead to different risks of dermatophyte infection. It stands to reason that we should find that. We indeed did. So that, and there's a small mistake. The p-value shouldn't be 10 to the minus 4, should be to the 14. But yes, so sometimes it's a lot easier. So basically what this is telling you is if you could target somehow keratin, you might have a chance to affect the risk of dermatophytosis. But still, the bottom line here is that things aren't always as easy, and a lot of times you need to do a lot of downstream analysis. And so this is the next step, the most technical part of the talk. And I think I'm a really big fan of this, of this approach that we use in genetic epidemiology. I think it's really revolutionized the way we work in genetic epidemiology. So I want to take a bit of time to talk about it. It's a method called Mendelian randomization, which, if you're familiar with the method called instrumental variable analysis in economics or in epidemiology, this is basically just this is just instrumental variable analysis, but using genetic variant as instruments. But before then, I show how we use it. I just want to take about 5 minutes to explain to you what it is, because I think it's actually changing the field right now in clinical epi. And to do this, thankfully, I'm going to take a little detour through cancer to explain to you what instrumental variable analysis is. And I didn't take this detour just because the crowd here is interested in cancer. This is actually my go-to example I give to everyone, no matter what their field is, because I think it's a beautiful example. It's actually one of the earliest example of the use of instrumental variable analysis in clinical epidemiology. So let's say you transport yourself back in the 1950s and you look at different registry data and you go, there seems to be an association between smoking and lung cancer. I wonder if it's causal. Nowadays, of course, we know it's the case, right? Saying the opposite would be complete nonsense. But if you want to be scientifically honest, in those days, you at least have to think of alternative hypotheses to this being causal. And the ones that were always brought up was, of course, there's other types of biases, but the bigger ones that we encounter often are confounding biases and reverse causation. So confounders for smoking and lung cancer, as for everything in epidemiology, socioeconomic status is probably the major confounder that we encounter. So unfortunately, if you're poorer, this is associated with more smoking and also associated with more lung cancer. So maybe it's just socioeconomic status that explains this. Reverse causation was actually used by a tobacco industry to explain that, no, actually smoking is good for you because the people who are predisposed to lung disease, including cancer, what they seek is relief, and smoking gives them relief. And so what you see is reverse causation. Smoking is actually good for you. Now, we know it's nonsense, but 70 years ago, you had to be scientifically honest and think, maybe this could be true. And so what early epidemiologists did is they used a method called instrumental variable analysis, where they said, if I could find a variable, which I'm going to call the instrumental variable or the instrument, that was associated to lung cancer only through its association through smoking, then with a bit of math, I could recapitulate the red arrow there, the causal effect of smoking on lung cancer. And the classic example, it's not foolproof. There are some assumptions that don't always apply, but the classic example in the literature is cigarette taxation rates in different jurisdictions. So if cigarette tax have an effect on lung cancer in a specific jurisdiction, then biologically it has to act through some other mechanism, and it would make sense that it would act on the risk of that jurisdiction population to smoke. So basically, higher cigarette taxes leads to less smoking, leads to lung cancer. Therefore, with a bit of math, you can recapitulate that smoking causes lung cancer. This is basically instrumental variable analysis, and this is basically what Mendelian randomization is. So basically here, instead of using lung cancer, would be using dermatophytes. Instead of using smoking, would be using gene expression, but it could be anything. It could be a protein level in the blood. It could be smoking. It could be anything, really. And instead of tobacco tax rates, it would be genetic variants that we find through the different GWAS, basically. Okay, so we have the GWAS for dermatophytes. We have GWAS for gene expression or gene level or whatever exposure you're interested into. And as long as you make sure that this genetic variant is only associated with, in this case, the dermatophyte through its effect on, in this case, gene expression, then you should be able to recapitulate this red arrow of the causal effect. I'm being very hand wavy here. And I'm not going to go through this in detail, but it is clear, I think, to anyone who's listened to everything I just said, that sometimes proving that there's no association between the genetic variants and the outcome, the dermatophyte, other than through the exposure gene expression, sometimes it's not that easy. But bear with me, at least for the purpose of this discussion. We tried our best to mitigate that. But yes, that is one of the major assumptions that we need to always check. Not going to spend too much time on this, but basically, if the assumption is met, you can do causal inference and presumably then identifying a gene that if you modulate its expression or modulate the protein level, it actually modulates the risk of dermatophytosis. And so that's what we did. I left the bottom part here where it's written colloq, just to the more genetics-minded people. If you don't want to look at it, it's completely fine. The results from Mendelian randomizations are at the top there, MR. Basically, what we did is we went methodically through every one of our 35, 40-something loci in the GWAS. We looked at all the genes that were within a certain interval. I think it was 500 kilobase pairs on each side of the lead variant. We looked at whether or not we had data on gene expression in the skin, so either sun-exposed skin or not sun-exposed skin. And to do this, we used the GTEC consortium data, if you've heard about it. And then we used Mendelian randomization to see if gene expression in the skin of those different genes was actually associated with the risk of dermatophytosis. We did other types of MR, but I think this is where the most interesting results are, so I'm just going to show them to you. And then what we noticed here, we tested a lot more genes than those, by the way, and we applied the FDR 5% correction because there was at least 250 genes that we tested. But basically, the ones where we could find something that passed this FDR correction are those ones. And so basically, if there's a little asterisk there, it passed the threshold. If there isn't, then either there's an NA, which means that this gene was maybe not expressed enough, or there wasn't enough data to actually perform the analysis, or there's a little square there, which means that it doesn't pass the multiple correction testing. But what we saw was actually quite interesting, is we saw a lot of skin related diseases, genes, so genes that are known to be involved in those different diseases of the skin, mainly psoriasis, eczema, but I forgot to put, but also acne was one of the ones that popped up quite a bit. Of course, in the immune system, it's an infection, so it stands to reason you're going to find some genes that are expressed in the skin that are related to the immune system and immune response. And what's interesting is we found genes, even though we didn't look at rare variants, rare pathogenic variants, we only looked at common variants that don't give very strong phenotypes, but some of them, especially a SLRP1, right there in the rare disease category there, still they still fell into these genes SLRP1 that are known to cause rare diseases when you have the wrong or the right, depending on your point of view, pathogenic rare variant. SLRP1, there's some families who suffer from a disease called mal de Meleda because they carry a very rare pathogenic variant. And that disease, mal de Meleda, is associated with hyperkeratinosis of the skin. So basically, they form a lot more keratin. And well, guess what? These patients, one of the main things that they suffer from is actually profound and difficult to get rid of dermatophyte infections because they produce so much keratin. And so all this to say, you can see that there's a few keywords that I dropped here and there about vitamin A and retinoic acid, right? So acne is treated with retinoic acid, psoriasis and eczema. Well, psoriasis can be treated. It's fallen out of favor because we have more effective treatment now. Eczema, actually, retinoic acid can either make it worse or better depending on the situation. SLRP1, the mal de Meleda, actually, we give retinoic acid, so vitamin A, to these patients to help with hyperkeratinization. And so we thought, are there genes that we found in our GWAS that are specifically associated with vitamin A, retinoic acid? Because we know how to target this very well using drugs. And so so this is just a slide because I was just to remind me to tell you what retinoic acid is. It's just vitamin A, but exists a ton of different vitamin A or retinoic acid derivative that you can use that are commercially available both in North America and in the UK. And so those are the two little genes that I'm going to highlight that are clearly associated with retinoic acid. So RARI, and the R doesn't stand for retinoic, it stands for some amino acid who I forget. But basically, here again, you have a locus zoom around the RARI gene. Here you see that the RARI gene is like massive, and that the GWAS signal at the top there falls right inside of it. So in those situations, it's easier to hypothesize that RARI is probably the causal gene, which we also see through Mendelian randomization and skin inspection. And this is the GWAS of skin expression in GTEC. As you can see, the red variants here, also the red variant there, they're both genome-wide significant, barely there, of course, in GTEC, but still a genome-wide significant in GTEC. And so, yeah, RARI, maybe I forgot to say, RARI basically is a protein that binds the retinoic acid receptor and modulates its activity. So we know it's clearly like it's a master regulator, as it's been called in some places in the literature, of the retinoic acid pathway. The other one that we found was FTO or RARIS2. Sorry, to be clear, they're not the same gene, but I'll explain to you. So basically, in the genome-wide association study, the GWAS for the risk of dermatophytosis, there was a very strong and narrow peak right dead smack in the FTO gene. Now, FTO, I'm sure some people in the crowd will remember that it is actually one of the first genes that they found as a risk factor for obesity. Now, obesity, we know very clearly, it's well documented and well established, and you can actually test it using Mendelian randomization. We know it's a strong risk factor for dermatophytosis, so that makes sense too. What's interesting, though, is that these variants, DED-SMAC and FTO, they are also strongly associated with circulating levels, so blood levels of the protein called RARS2. And in this case, RA does stand for retinoic acid, so retinoic acid responsive element, something like that. And it's basically a protein that is upregulated as you give more vitamin A, basically. And what's interesting is that we know that obesity leads to a relative state of deficiency of vitamin A. And we know, and this is really not the clincher, but I thought was very interesting, and I'm going to say to you because some of you guys probably work in this field in oncology, but we know that one of the genes that is actually upregulated in cases of acute promyelocytic leukemia is FTO. And how do you treat acute promyelocytic leukemia? By giving retinoic acid, vitamin A. This is strangely the treatment for one of the most quickly lethal forms of leukemia. And the prognosis is excellent once you start the treatment. Now, I'm not saying that because we find something in leukemia, it's going to help us with dermatophyte, but I just thought that, like, the constellation of finding was too cool not to tell you. So basically, yeah, we have like quite a bit of evidence there through our MR with other skin-related genes, the MR with rare disease-related genes, even though it's true common variant and not rare variant. And now these two genes that we find using Mendelian randomization. And so, yeah, we went back to our patient, and our patient was really smart, college-educated guy. I actually presented to him our findings a bit like I presented them to you today. And I asked him, listen, like, are you convinced? Is this something you want to try? Basically giving isotretinoin, which in US and Canada is called Accutane. And I think in the UK, I double checked, I think it's just Roaccutane, which is very frequently used for acne and works very well. And so he said, yes, let's try that. And within about a week or two, he noticed very rapid improvement in pruritus and discomfort. And I'll show you the picture. Now, the picture is a bit of a caveat. The caveat is that on the left, it was taken the summer, so he had a bit of a tan, and on the right, well, it was in the middle of the winter, which, as you can imagine, in Montreal, we don't really go out in the sun. So it probably amplifies the difference there. But what you can't appreciate maybe too well is that the scaliness is basically gone at this point. And of course, you can't appreciate this because it's a subjective symptom. The patient said the pruritus was gone. He was basically back to having normal, smooth skin. It was still a bit discolored. You can see a bit of a border there, but it was clearly better, even correcting for the tan that he lost over the winter. So yeah, this was kind of the journey we went through with this patient, and this got us to think, has there been other work done on this? As far as I know, I'm not aware of anything that's been published that basically went from patient to genetic epidemiology back to patient like we did, but we're not definitely the first to think about like polygenic risk, common variants affecting the risk of developing diseases that are more monogenic. Some of the earliest work I could find was actually in 2020. I'm not entirely sure what this group only looked at: coronary heart disease, breast cancer, and colon cancer. And I'll show you after their figure for colon cancer. They did find that yes, the polygenic risk of common variant does affect your risk of developing colon cancer, even if you carry an allele that is causal for Lynch syndrome. They found similar results in schizophrenia, and then also recently in Nature Genetics and developmental disorders. And they used like educational attainment to measure developmental disorders as well as other measurements. But I think, there's a sense now in the field that as if we go back to the previous figure that I showed you, we're all on a spectrum basically in terms of risk. And some of it is modulated by more rare variants and some of it is modulated by more common variants. And the point is that our methods of genetic epidemiology work really for different reasons, but they really work best in the common variant field, and you can translate that to a treatment. Well, hopefully you can also treat even the carriers of rare variant monogenic diseases. And just to give you, I'm sorry, it's a bit blurry, but this is basically the figure in that paper with colon cancer. On the left there, you basically have the odds ratios, and on the right, you have the probability of developing colorectal cancer by the age of 75 years. In blue, the carriers of Lynch syndrome variant. And in black, those who are non-carriers. So the non-carriers basically have their only risk genetically is polygenic. So basically from all the sum of all of those small betas, and if you're unlucky enough, you have a very high sum, and therefore you have a higher risk. But the point is, yes, of course, Lynch syndrome variant, they're going to have a higher risk, a higher lifetime risk of developing colorectal cancer, no matter where they fall within the spectrum of polygenic disease risk variance. But clearly, those people who have a lot of small effect size protective common variant allele, even if they carry Lynch syndrome variant, they have a much lower risk than those who carry a stronger polygenic risk. And that's kind of, I think, where this field is going. I think this is very interesting because, as I said, the black line there right now, it's very active in terms of translation from biobank scale data to novel treatment. And hopefully we can use that in those Lynch syndrome carrier, for example, to mimic a polygenic state of protection and push them back on the left side there to a lower risk. Of course, higher than the population, but hey, you know, that's still better. But that actually kind of answers this question from my conclusion is where to from here. I think the real answer will be it depends on the disease. You know, I think in cancer, I think you guys would be more interested in this case of like preventing cancer than treating cancer, or as an infectious disease, it's probably more going to be more about the treatment. But yes, I think there's some work to be done there. We've started to look at the UK Biobank and rare disease variant carriers. For example, there's a lot of people with macular degeneration, and there's some very good polygenic scores and GWAS for macular degeneration. So we could easily check if there's something we can do to help those people where rare monogenic disorders affecting macular degeneration. But yeah, that's basically what I have to say. So if there's any question, I'm all yours.